Namaste students. I am JV Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic various downstream processing steps. So what are the steps which are involved in the downstream processing subject? And the topic belongs to the subject downstream processing in the BSc Biotech. So first, before going to the various steps of the downstream processing, we need to understand about the downstream processing. What are the downstream processing and what are the upstream processing? And also, we need to know that which step we are going to do. So, in this video lecture, we are going to study about the first step of the downstream processing. So, first, we need to understand what is the upstream processing. In the industrial microbiological experiment, we perform all these experiments for the production of enzymes, vitamins, some special compounds from the microorganisms, so that we can isolate them, we can purify them. So for production of these uh, proteins and enzymes, we need some equipments such as we need some bioreactors, we need some other enzymes, we need some growth media, we need some growth factors, we need some optimum conditions such as pH, temperature. Okay, so preparation of any kind of reaction is will be considered in the upstream processing. Okay, it is the preparatory step by for which we are going to study the fermentation step. We are going to prepare for the fermentation, so that will be known as the upstream processing. Now, what is will be the downstream processing? Various stages of processing that occur after the completion of the fermentation or bioconversion stage, including separation, purification, packaging of the product, is known as the downstream processing. So, suppose that we need to prepare some enzyme, lipase enzyme from the microorganism. So, after fermentation process, we need to first uh, separate the enzymes. We need to separate the microbases, and then we need to separate the enzymes. We need to then purify the enzymes, and then after purification, the enzymes must be packaged. Similarly, we need to prepare some antibiotics. Okay, from microorganism. So for micro antibiotic, we need first take all the preparations, then we separate them, we then purify them, then after purification we go for the crystallization steps. So these all steps are will be considered as the downstream processing. Means it is it is the process which occurs after fermentation, the original after the original steps. So what are the downstream processing stages? So various steps are involved in the downstream processing, which involves the first removal of the insolubles. The insoluble particles will be separated. Then product isolation, the second step. Then product purification, the third step. Then product polishing. Then the sub product is transferred into the market. So these are the steps. Removal of insoluble, product isolation, product purification, product polishing, and A product recovery methods are considered to combine most things. The examples include expanded weight adsorption, complex removal of insolubles, and product isolation in a single step. Affinity chromatography often isolates and purifies in another steps. So this is the example. Now we go to the first step of the downstream processing, which is the removal of the insolubles. What is the first step? It is removal of the insolubles. The separation of cells, cell debris, or their particular matter means they are not soluble parts. They can be easily separated. So this is very easy step because in this step some insoluble compounds are separated which which are not soluble in the product, and the typical operations which are used in this step such as the filtration, centrifugation, sedimentation, flocculation, gravity setting. So these are the steps. So we are going to discuss about all these. First is filtration. What is the filtration? Filtration is the mechanical operation which is used for the separation of solids from the fluids. So it is simply sieving a water sample. Suppose there is some insoluble compounds or waste material will be there. So what we do, we take a sieve and we filter the water, and the solid material will be separated out. Simple thing is followed in the filtration step for the downstream processing layer, in which we use a sieve. Okay, so in this step we use a porous membrane through which the fluid will pass and the solid material will be retained on the upside position. They will not be flown through the membrane because of their larger size, and the liquid material has lower size, more size, so they will be filtered. They will be separated. The solid particle deposit on the filter will form a layer, and which is known as the filter cake. Suppose that we are making we are adding different cells and our samples there. Okay, then what happens? The liquid particle will flow through the membrane, but the solid particles, which contains the cells, okay, which contains the microbial cells, will not be separated because of their large size. So they will be in the upside position, okay, and they will form cake-like structure, which is known as the filter cake. Okay, they will not be filtered and they will be uh, in the upside position of the membrane, and this will make it cake formation. All the solid particles formed by stored by cake, and the cake grows at the rate of which particles are brought to the surface, and all the fluid goes through the cake and filter medium. So this is about filtration. Now centrifugation. Centrifugation is the next step. Centrifugation is used to separate particles of size of 100 to 0.1 micrometer, okay? And they are separated from liquid by gravitational forces. It depends on the particle size, density difference between their cells, and their growth and their viscosity. They are more viscous if they vary in the size, they will be separated easily. And in this method, we use centrifugal force for the separation of the mixtures, and more dense components migrate away from the axis of the centrifuge, and less dense components migrate towards the axis. Types of centrifuge use are tubular wall centrifuge, multi-chamber centrifuge, and the disc wall centrifuge, for which we will discuss in upcoming lectures. The next step is sedimentation. Sedimentation. What is the sedimentation? It is possible for only for large particles greater than 100 micrometer flux. It is for the large particles greater than 100 micrometer. It is a slow process and takes about 3 hours. It is used in the process like activated sludge effluent treatment. It is a, so sludge is a different level of experiment because they have larger size. The particle size is larger in the sludge, and it's a free settling process depends on the gravity. A particle settling is a highly particle density suspension. It's a very simple process. The next is flocculation. Flocculation is a process where a solute comes out of the solution in the form of flocks or flakes. So what happens actually in the flocculation? There are some flocks are formed, and we can also say that flakes are formed in the flocculation. In these particles finer than 0.1 micrometer in water remain continuously in the motion due to electrostatic charge, which causes them to repel each other. Means when the electrostatic charge is provided to the particles, then these this causes them to repel to each other. Okay, they will repel each other. They will uh, not allow themselves to come together. They will repel each other. Once the electrostatic charge is neutralized, then the finer particles will start to collide and combine together. And the larger particle will be on the other part. Okay, the larger heavy particle will be another stage, which will be flux. The smaller particle will start to collide and combine together, and the larger particle or heavy particle will also start to make the uh, collides, which are known as the flux. Okay, so they will be separated from uh, each other by this process. That they need to be separated by their particle size. The small particles will be uh, adhered in one place, and the larger particle will be adhered on the another place. So this is all about the downstream processing step one. So I hope that students have better understood what is the downstream processing step number one. What students will learn about the downstream processing step number one. The students will be able to understand and explain about some steps which are involved in the downstream processing, such as filtration, sedimentation, centrifugation, flocculation. So they will explain this very well, and they will also explain the introduction of the downstream processing and also about the introduction of the upstream processing. So what kind of questions can be asked? The questions which can be asked is explain downstream processing, explain the process steps which are involved in downstream processing, explain the first step in the downstream processing. So these questions can be asked. So my lecture is over here. You can ask me questions. Thank you very much.